follow.
Oh, was it? I see. And it's a little bit outside of what we've been reading so far, but we have talked about like hibernation and things like that. I thought it might be interesting. Also, I just like how thorough. I thought the writing was pretty clear in the sense that they said, this is what the experiment we're going to do, and then boom, 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 do the experiments, and then say, like, because of these results, we conclude that these are happening in glutamatergic neurons, blah, 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 right? So I thought the reading, for how concise it is pretty clear, as clear as they could make it to be. I mean, the biggest issue is how is violet light detected, right?
Yeah.
Okay, so let me share the page. Oh, just an announcement. Next week I'm going to post a paper from bioRxiv about 3-photon imaging. Nobody's going to understand it, me included, because it's all about like laser properties, how to measure them, laser dispersion. But there's a laser core here, and an expert who specializes in laser properties. So I asked him to come to our journal club and present. So he'll give us like a primer on lasers, optics. So I'll post.
So we're not presenting, we just read it ahead of time and then they'll present to us.
That's the goal.
Right.
It's like gibberish. It's hard to understand because optics wasn't something that I covered in college. So learning things in college matters, Amanda. It's not too late for you. Yeah. All right, so let me share this screen. Let me get rid of this. Is this good?
Yep.
Okay. I guess the background is pretty clear, right? They detected opsins in a brain region, the hypothalamus, that is potentially involved in thermal regulation. They wanted to see if light could directly stimulate, or whether these neurons could directly detect light and what effect that would have on thermal regulation. So they focused in on opsin 5, and Amanda will tell us what the opsin 5 expression looks like.
Yeah, so essentially they believe that opsin 5 is a violet light sensitive, it's an opsin, right?
Yeah.
That's what it is. It's a protein. Okay. Which aids in the thermoregulation of mice. And so opsin 5 resides in both the retina, but also the POA, which is a thermoregulatory region that controls the BAT, which I looked up is brown adipose tissue, neuraxis, in the hypothalamus. And so figure A and B was they used a knock-in allele that activated DT tomato. So they identified opsin 5 expression in the POA. So that's just confirming that it's present. Figures C and D show that this region transcribes opsin 5, and they used XGAL labeling, which is galactose with indole, and it's a good gene reporter. And there's no opsin 5, they noted, in brown and white adipose tissue, the thyroid, liver, heart, adrenal glands, or pancreas. POA has many neuron subtypes that are involved in homeostasis, and so fluorescence was used to highlight those subpopulations, and that's the rest of this figure. So most opsin 5 also expressed SLCA6, which is glutaminergic, which means it's excitatory. And a little bit contained SLC32A2, which has a GABA exporter, so that's inhibitory. And so F through H just shows that it has mostly excitatory receptors and genes. And then there are also temperature-sensitive neurons that contain PACAP, which is pituitary adenylate cyclase activating enzyme, and BDNF, brain-derived neurotropic factor. So I looked all those up because they didn't really give us the definitions. And then they use TRPM2 as a heat sensor. And so they found that almost all opsin 5 POA had both BDNF and ADCYAP1, and half also had TRPM2. So thus the opsin 5 neurons use these receptors as a way to be warm sensitive and glutaminergic. And this segues into the next segment where they want to see how opsin 5 is related to BAT to control thermoregulation.
Very good, thank you. So from E to K, what molecule of opsin 5 are they measuring through this technique?
They did like a knock-in with Cree, if that's what you're trying to get at.
From E to K.
Oh.
These colorful images.
It's the opsin neurons, right?
Yes, right. But what are they detecting here? Using
fish, right?
Fish detect
Genes?
mRNA.
Like the genetic makeup of the protein?
of the mRNA. Yeah. So
But opsin 5 is a protein, so that's where I was getting a little confused. Is like these are genes. I wasn't sure if they were the genes that are transcribed into the protein or a receptor.
That's where I was getting into. So
okay.
Let's start from the E to K. So this is a method of using fluorescence, and it's a fluorescent probe, essentially. A fluorescent probe an antisense probe that will bind to mRNA. It's actually a tandem of small probes, let's say a collection of 50 probes that bind specifically to mRNA of interest. So you would buy this tube with these fluorescently labeled antisense probes against opsin 5, VGLUT2, BDNF, right? Each one with a different color, and then you would mix them together. And if the cells express BDNF, let's look at K BDNF. You see all these small dots, right?
Mm-hmm.
Each presumably would be a molecule of mRNA. So those many dots, we can kind of say that these are all the mRNA molecules of BDNF that are present in this one neuron here. And the other dots for, let's say, ADCYAP, those are all mRNA for ADCYAP, labeled in blue. And this method, which has been developed in the past 10, 20 years, is so sensitive that it almost gives you single molecule resolution. You can detect each individual mRNA. Not completely true, but that's why they're all spotty, like dotted. So here you're measuring mRNA expression. But actually they cheated a little bit. They didn't do mRNA against opsin 5, but they're doing mRNA against TD tomato, which is expressed by Cree. I don't know why, because they really should have just done opsin 5, BDNF individually, and they should be able to detect it. It might be that opsin 5 expression is not very strong, or perhaps a lot of the mRNA is not in the cell bodies. Maybe it's in somewhere in the processes, who knows, right? But
Mm.
that's something that they didn't explicitly say, but they cheated a little bit, and they're looking at reporter.
So they're using the TD tomato to represent the opsin 5?
That's right.
But how did they know that they would correlate / how did they do that?
Because opsin 5 Cree would be expressed in cells that express opsin 5, and with that, using that Cree, they're expressing TD tomato.
Okay, thank
Right.
you.
All right, so then having said that, I wanted to step back a little bit and look at A, B, C, and D, right? Does everyone know what this superscript means? When you have a Cree superscript / plus, what does that mean? And it comes up like multiple times in this paper. So this means that it's a knock-in, most likely.
Yeah,
Where
I asked you about that because I wasn't really
the
sure.
Cree is in the opsin 5 locus. I didn't read this carefully enough to know if the Cree replaced, this replaces the gene actually, right? In this paper. The Cree replaces the original opsin 5 gene. So in this mouse, opsin 5 Cree / plus, the plus means that it wild type, right? The original gene. So this means that in this mouse, there are two alleles, the two arms of the chromosome. One has the original opsin 5 gene, that's the plus, and then the other has the Cree instead of the opsin 5. Make sense?
Yep.
So let's say you cross this opsin 5 Cree / plus, so opsin 5 Cree heterozygote, to another mouse that has opsin 5 Cree plus. Right? Then in 25% of the pups, you're going to get plus, so wild type animals.
Wild type.
50% you're going to get Cree plus, and then 50% you're gonna get Cree, 25% you're gonna get Cree, which would essentially be a mouse that doesn't have opsin 5 anymore. It's making Cree instead.
Yeah, I think they called that like opsin null.
That's right, because they're making Cree instead of opsin 5.
Yeah.
So same deal with CND. They're using opsin 5 LAC-Z. So LAC-Z would replace the opsin 5 in one of the chromosomes. Yeah. All right. And then a little more into mouse genetics. I think by now we're aware that Cree is a recombinase, and that will recombine the LOXP sites. So in AI14, which is a TD tomato reporter line, in a cell that has turned on Cree at any point in its life of that cell, if Cree turned on even for a short bit, that will chop out the stop codons in AI14. So now that cell, even if it doesn't express opsin 5 now, is forever labeled red. Right? That's what they mean by lineage labeling, that at some point in that lifetime of that cell, it turned on Cree and it excised out the stop codon in AI14. So now that cell has red forever. Yeah, makes sense?
Yep.
All right. So the difference between that one and opsin C and D is that A and B labels tissues forever, but LAC-Z and C and D is only turned on when opsin 5 promoter is on, right? So that's the difference between the Cree lines and the LAC-Z lines.
Is it as powerful as Cree?
Not as powerful, most likely, because this is dependent on, so this Cree AI 14, what dictates the expression of TD tomato? What's the promoter for the TD tomato?
The promoter is the CIG, like
It's an artificial
artificial
promoter, not artificial, but it's from chicken beta actin, which apparently expresses at very high levels. Right? It's a very strong promoter. So in the mouse A and B, regardless of how much opsin is made, because the Cree has done its job, this red expression is not dependent on Cree. The expression level of red is dependent on the CAG promoter, as Zhang pointed out. So it's an optimized reporter where the promoter is very strong. It has a strong enhancer. It has other elements that stabilizes mRNA, so that TD tomato can be expressed at very high levels. And that's why it's so bright. But in the case of the LAC-Z mouse, this LAC-Z expression is dependent on opsin 5. So if opsin 5 is not expressed very high, it won't be detectable. But however, the reason that they use this very old technique of doing this blue labeling is because this is an enzymatic reaction that is pretty strong. And it can amplify signals. So even if there's not that many molecules of LAC-Z being made, it's still detectable using this method. And that's why instead of Alex presented on Tuesday about putting in a reporter, we saw how iffy that fluorescence was, right? So we have a bitter receptor Cree, which we can do bitter receptor Cree with AI14, same kind of strong expression. But our bitter red was not very strong. And they might have had the same problem. If they had put in OPN5 M scarlet, maybe they don't see it here in this expression. If we had made a T2R5 bitter receptor LAC-Z, we probably could see it much better than what we're seeing right now. However, we're banking on fact sorter to help us out. We'll see. Yeah, is this all making sense? Yeah. All right. So then, who will go over?
I'll do figure 2.
Figure 2. Yeah.
Okay. So let me pull my notes. So now that they identified opsin 5, they wanted to see if they are connected to the brown adipose tissue. So to do that, they injected transneural retrograde pseudorabies at the brown adipose tissue. And then with the mechanism of these rabies, is going to trace backwards to all of the various areas in the brain that all converge onto the BAT. Mm-hmm. And so from figures B to I is just the red dots show these neuron populations have the MRFP1. So in B, it is the intermediolateral nucleus of the spinal cord. C is the raphe pallidus. D is the dorsal medial hypothalamus. E is the paraventricular nucleus. F is the NTS. G is the lateral hypothalamic area. And H and I, the areas that the authors are more interested in, it's the POA. And basically, this result shows that there is a direct polysynaptic pathway connection between the opsin 5 and the POA and the brown adipose tissue.
I have a question.
Yeah.
Can we hear me?
Yes.
Yes.
Oh, sorry. They're claiming that there's direct polysynaptic connectivity, but I thought pseudorabies was a monosynaptic like marker or whatever. Like it only transfers one synapse back. Are they saying that there's like two neurons that are, you know what I mean? Like I'm kind of unsure of what they're claiming.
Yeah. So monosynaptic rabies usually are G protein deleted. Because the rabies have G protein deleted, so when it goes one step further, the second cell, the target cell, will not have G proteins, so they cannot propagate further. But here it is only pseudo rabies, but it's not G protein deleted, so it only have a pseudotype envelope to target the starter cells, but they will be going further.
That makes much more sense.
Also, although this is called pseudorabies virus, this is more of a herpes family of viruses, so not the rabies family. And this one does retro, but it can jump multiple times. And
I thought HS herpes was antero. Is that wrong?
the HSV that was discussed before is antero.
Yeah.
Yeah, but this is a retro version. The pseudorabies family goes retro. Yeah, lots of viruses.
Engineering.
Yeah, but as you can imagine, these neurons or this virus jumping over several neurons, it's very toxic to the animal. So the problem of using pseudorabies PRV methods is that over time, you'll see lesions happening in the earlier tissues or the earlier neurons. And I think typically they also time it so that after day one, it goes one station, let's say. I'm making this up. Like 3 days later, it might have traveled like 3 stations and so forth. Jahal, where's this NTS bit? This NTS, the NTS.
The NTS is almost at the quarter part.
Yeah, what
You will have mostly it's in the dorsal motor vagus.
right. This is not really the NTS region that gets the vagus input, right? This is more the motor.
Yeah, motor part.
Yeah.
That's true.
Which is expected. I would think there might be, the DMV might, DMN might be synapsing onto the BAT. All right. Okay, I think we can go on, John.
Right. So after they show the direct connectivity, they then wanted to show the mechanism using a DREADS approach. So they injected the excitatory HM3DQ or inhibitory HM4DI designer receptors using Cre-dependent viral vectors into the OPN5 Cre/plus mice and compared it to the wild type mice as control. So in J is just the experiment design. K on the left side is the Cre, so it shows that the Cre heterozygous mice have the M cherry, so they have the receptors, while the wild type I do not have any of these DREAD receptors. And then M is the timeline for the experiment. So they injected in week 5. 2 weeks later, they added a telemetric sensor to monitor the brown adipose tissue and the core temperature. Temperatures there. And by a couple of weeks later, they injected with CNO to activate these receptors, either excite them or inhibit them, and they saw the results. So at the bottom, on the left side, left two pictures, it's the excitatory. And so N and O, this is with the Cre receptors. So activating these opsin neurons led to a significant decrease in both the brown adipose tissue as well as the core body temperature. And the control is the bottom two figures, P and Q. And on the right side is with the inhibitory receptors. So inhibiting the opsin receptors led to a significant increase in both the BAT temperature and the core temperature compared to the control.
I have a quick question. I know you went over this, but the plus is the wild type, and then the Cre plus is the one allele with Cre, but I was wondering like what that does exactly, having one allele be Cre versus the wild type. How does that affect the opsin?
Good point. And so here they're injecting the GQ and the GI viruses, which are Cre dependent. Right? So if they were to inject, let's take the GQ example. When they inject this GQ virus into the Cre animal, this GQ would be expressed because of the Cre in the opsin 5 expressing neurons in the POA, right? Whereas they'll do that same experiment of injecting GQ into the opsin 5++, which doesn't express any Cre. So in that mouse, GQ will not be expressed.
Are they just confirming that the Cre works, or are they trying to actually test something?
Well, they're testing the effects of activating opsin 5 neurons using this DREAD technology.
Oh,
right?
Got it. So they activated just the opsin 5 because it has Cre.
That's right.
Got it.
So this is a good way to show control. Here, what they're doing is they're doing the same surgery of injecting viruses, and then they're injecting CNO, but they're comparing the Cre animal that would express DREAD in opsin 5 neurons in the POA versus animals that don't have the Cre, so that they won't express GQ. So if there are any side effects of CNO, any side effects of this virus, then they'll see that.
So they're just like targeting, they're only activating opsin 5 with the Cre, and so they know they can see that like direct relationship. Okay, thank you.
Activating neurons that express opsin 5.
Yeah.
Right? Not the protein itself.
I have a question. Why didn't they show me a picture of the opsin 5 Cre cells? Like, they're showing them to me in A. I would like to see these cells double labeled. You know what I mean? I would like to see the opsin Cre in green and the M cherry.
To confirm that they can inject in the correct site?
Yes.
I would assume that that most people will just take their word for it.
I'm just like, I see very
especially because they're seeing very strong phenotype. Yeah. It might be in one of the supplemental figures, which I didn't dig into. Yeah. Okay. So did we discuss the effects of activating these cells and inhibiting these cells?
Yeah, so again, activating these cells leads to a decrease in both the brown adipose tissue and body core temperature, while inhibiting these leads to an increase in both the BAT and core temperature.
Yeah.
So these neuron populations can bidirectionally regulate the brown adipose tissue activity.
Mm-hmm. All right. Well, so far it seems that I'll go into this in the next figure. Yeah. So, figure 3. Anyone? Takers for figure 3?
I can take it. Figure 3, they are just trying to test if they actually apply the violet light, if that will give a difference in the temperature change. So A, B, and C, D. So C and D is without violet. You can see on the left there is a lacking of 380 nanometer light applied. So they are the controls. And A and B responds to the bed temperature and core temperature respectively with the violet light. So in A, so since the violet light is applied between hour 3 and 5, so that is when the changes start, you can see the dotted line there. If we see C and D as negative control, there's really no difference in the temperature change that's represented by the red line they drew. But for A and B, if they apply the violet light, they can see a significant decrease in the option 5 positive mouse line, where they didn't observe any decrease or like significant decrease in the negative ones. So they conclude applying the violet light. So their conclusion here is the option 5 is responsible in its function of receiving the light in regulating the temperature. And for E to H, that's raised by another concern is that because the violet light, they didn't shine it directly in the brain, but they just provided it to the mice, then opsin 5 is also expressed in the retina, in the eye. So how do they know it is not the retinal opsin 5, but the brain of opsin 5 that regulate the temperature? So so in E, they just use a Cray line to remove the opsin 5 expression in the retina. And they see there's no difference in the temperature regulation. So they further confirm that the retinal opsin 5 is not playing the role, but rather it is the brain opsin 5 or like the POA ones. And in F to H, I'm a little bit confused with the enucleated mice because enucleated, to my understanding, is removal of the nucleus here. But I don't quite get that part.
In the eye, no?
It's still in the eye?
Yeah.
Okay, so it's the same wait, is it in the eye? So that means they are also they're not removing it from the eye again, right? This time they observe a difference. I'm a little confused on this one. Let's see. Someone could jump in. I mean, for H, it is a qPCR test. Like they just tested several genes that are shown to be functioning in thermogenesis, and they found that in the negative one or in the opsin 5 positive one in H, you can see there's much lower expression level of these RNAs. So that means the thermogenesis, these genes are actually regulated by the expression of opsin 5. But for F and G,
no, so in E, what they're doing is they're removing opsin 5 genes from the retina, right? So the retina is still there, and they're seeing the effects of opsin 5 gene or protein not being made in the retina, right? And there's no difference. In F and G, they're removing the eye.
Okay.
These are eyeless blind animals that that you see a difference, right?
Yes. Oh, okay, I see.
Wow.
So removing the eye will result in a temperature a difference, right?
So it's a much, let's see. This would suggest that opsin 5 is doing something even in the absence of the eye, right?
Okay.
So the light is not really coming through the eyes, but opsin 5 is still able to detect violet light, and yeah, it's able to detect violet light in G from somewhere. We don't know where.
But doesn't it conflict with E because they're
No,
only different?
so here they're saying that the opsin 5 in the POA is important, but the opsin 5 expression in the retina is unnecessary for this. Right? This is a specific expression. So does everyone know the difference between opsin 5 minus minus and what they did here?
What I'm confused about is why are they not seeing the same effect using the retinal deletion of opsin 5 versus enucleated mice? Like, isn't that in theory doing kind of the same thing? You're removing opsin 5 from the retina, shining the violet light, and then you're removing the eye and shining violet light, and they see a larger effect when you remove the eye. That's why I'm wondering why I don't.
No, but in E, they're just removing this one gene.
Yes.
Yeah, and in the eye, in the retina, and doesn't seem to have much effect. The case that they're trying to make is that opsin 5 is very important for this, but not in the eye. It's detected through the POA somehow.
This is just a guess, but is the opsin 5 minus minus? There's no opsin in the POA either. And
That's right. In the opsin 5
Oh, I see.
minus minus, opsin's not there in any cell in the body.
Yeah.
It's a knockout.
Yeah, all the opsin 5s are gone.
Then this is just the eye.
And E is just in the eye. So how they do this is that flux FL, that means that the LOXP, which is target for Cree, is flanking, is in the either side of the gene, so that if there's Cree expression, the recombination will happen and that will chop out the gene. Right? So they used a driver, the RX Cree is expressed in the retina. So specifically in the retina, opsin 5 will be deleted, but the POA opsin 5 is still there. And so then E suggests that the POA or whatever other non-retina opsin 5 might be doing this according to the later figures, but at least when you remove it in the opsin 5, they don't change anything, because you see that decrease starting from time 0. So take-home message for that one is retina opsin 5 unnecessary.
Yep.
At least for this effect. Yeah. It might be doing other things, but not for this.
Is it explained at any point why the core temp is starting at the different point for the different groups of mice? So why do opsin plus start at 37-ish and the knockouts are, at 38, 9?
They already can't regulate.
So it's always higher.
Yes, because this, we saw that in the earlier figures, right?
Mm.
Here. So,
So the baseline is different.
Yes, because this in the absence of opsin 5, you're getting heat production, more heat production.
Okay.
So they already lost control of their heat.
Yeah.
Heat generation.
But yeah.
So having different baseline for the subjects might also affect the experiments. So I don't know, maybe they could do conditional knockout using maybe tamoxifen to show that the difference from the same baseline.
Very good. So I think it might be in one of the extended figures, but they did an experiment where they were concerned that here they're removing opsin 5 from throughout the body, and it could be that opsin 5 is expressed, say, in the tail, and that's how they know the temperature or the light, and that regulates temperature, right? So they mentioned that they did an experiment of using leptin receptor that is expressed just in the POA or other places too, but mostly in the POA, to specifically remove opsin 5 from the POA. And they say that they see the same effects. So it's in fact opsin 5 expression in the POA that has this effect. But as Alex pointed out, that's a great experiment to do, where you can make leptin receptor Cre-ER, which is dependent on the presence of this ligand tamoxifen. So you could have, in this mouse, the removal of opsin 5. So if it was a leptin, this is going to be very complicated. I wanted to use chat, but I don't know how to use chat. Oh, here. So that would be leptin receptor Cre-ER cross to optin 5 flox, right? Like this.
Yeah.
So in this case, Cre will be expressed in the leptin receptors, but the Cre is floating around in the cytoplasm, not able to go into the nucleus to do the recombination. You inject tamoxifen in these animals, then the Cre will finally go in, flux out opsin 5, and then in those animals, you would see, like in figure 3A, the blue line, right? No, not the blue line. It'll be the gray line.
Yeah.
And then when you inject tamoxifen, then maybe the temperature goes up, and now you finally go closer to the blue line.
Yep.
Yep. That would be the ideal situation. But you can imagine that this recombination has to happen in both arms, right? So it's not going to happen in every single opsin expressing neuron. So the efficiency is pretty low. And that's why it's hard to do those kind of experiments, although that's definitely the experiment that they should do, because they'll see then a switch from normal mice-like behaviors to more body temperature, higher body temperature. Yeah. All right, is this all sort of kind of making sense? So it seems that in F, there, even if the I isn't there, that your opsin 5 is regulating body temperature, and that seems to be dependent on violet light. Yeah? Good so far? All right. So who'll go over figure 4?
No one. Okay.
Yeah, how?
So
You're the imaging expert to be.
anyway, so
Yeah.
okay. Based on this promise,
Yeah.
let's see how this figure does.
Yeah.
So they shows us those opsin 5 expressing cells in the PLA response to violet light, and then regulate the body temperature, like core body temperature, but also the brown tissue adopts the adipose tissues, so lipid tissues. So then there's a later question to ask: how does this happen? Because if you have light, so those are photons, you need, maybe there's some mechanism to mediate the effects at the neuronal level. And what they do, they start cutting the brain into the slice, and you have the chrono slice in A, and in these like in vitro preparation, they can both monitor the temperature of the solution, and then at the same time using the 2-photon imaging techniques called FRET, basically relies on 2 different fluorescence proteins. One has a slightly longer emission of fluorescent light with a YFP, like yellow color. One with a slightly shorter wavelength in the cyan, so CFP, cyan meaning like blue light. So basically when there is when these, I think it's conjugated protein, get closer each other, like in the bottom right of figure A, they will emit the yellow fluorescent protein mainly. While if they are in relaxed configuration, they mainly relies on emit the blue light. So that's the arrow of let's say CFP have an upward arrow, while YFP have a downward arrow at the relaxed mode, and then when get together it's reversed. And this somehow depends on the cyclin AMP. So if anyone remember molecular biology, how the GPCR works, one of its downstream messaging downstream signal is the cyclin AMP. And how does, also I forget to tell, so this is a mice that is a reporter mice that indicates that encodes both CFP and YFP, and can be used as a cyclin AMP XA indicator. So the changes of these 2 different wavelengths, fluorescence wavelengths, can indicate the changes of the cyclin AMP concentration. That's how all those figures based on. And then they start showing us in B that's what it looks like. I have to see what the condition for stimulation. B is
Violet light.
Huh?
Isn't it violet light?
Violet light, yeah.
Just violet light?
Yeah.
Okay. So, oh yes. I kind of forget a little bit detached.
So just for a little bit of big picture, naturally after the first 3 figures, we want to figure out how POA neurons are getting the violet light and whether they react directly to violet light, right? And from what we've been doing for the past couple of weeks, reading, you could think, oh, maybe we put GCamp in there, do photometry in the POA, opsin 5 neurons, and then just put the animal under violet light, maybe you'll see an activity. That they must have done this, right? Even though they don't say it, they must have done this. But probably didn't work out. I don't know why. So they still have this task at hand where they have to show that the opsin 5 neurons, POA neurons, are reacting to violet light. So they come up with this very elaborate experiment in a sliced brain to show that violet light increases cyclic AMP, and thus they are active. Is it convincing?
So yeah.
So what's not convincing for me?
Yeah.
Oh, it's probably yes, I would say.
It's convincing
because they did have this opsin 5 mouse line. Not sure but all of them have the opsin 5. They don't have an opsin knockout results.
I might have just missed this,
right.
but
Yeah.
Cyclic AMP is everywhere, so how did they localize like where exactly they were trying to detect cyclic AMP?
Oh, very good. So here they're using the opsin 5 Cree animal. So you can look at G, and the reporter, the cyclic AMP reporter is Cree dependent.
Yeah.
So
Got
then
it. Okay.
you would have the expression in Cree mouse. Wait, actually G, Jiahao. So here they're using the Cree to express camper, this reporter, in opsin 5
Oh, they do
right?
have, they
And
do
then
have
they
the
compare
control.
the presence and absence of the opsin 5 gene.
Yeah.
Because the gray line has the Cree in one arm, one allele, and then the functional opsin 5 in the other allele. Whereas the blue line is Cree, so knockout there, and a null there, and then the knockout in the other arm, that's the minus. So this is a homozygous knockout, in effect. And then that brain is insensitive to violet light. Right?
Yeah.
So still like if following the regular logic is overall like in vivo testing more convincing than ex vivo?
I mean,
No, you don't need that.
I think this shows that these neurons are reacting to violet light, but how does violet light get there in the first place?
Exactly.
Yeah, in an animal, it's not going to go. Do you know how deep this is, right?
Yeah.
This is at the very bottom, as you can see, it's at the very bottom of the brain.
They have the experiment actually to measure the light in the POA.
How much light gets in there?
In their supplementary figure.
Yeah.
How much? Which figure?
And violet.
I think the not. Yeah, the not
is
figure.
short.
So figure 10, they show the. Oh my god. They have implanted a micro probe, inserted until the below the cortical surface 4 millimeters, and you have the in figure D and E of that,
4 millimeters. So that would probably get to the POA, right?
Yeah, they actually physically measure the light intensity in the POA. Yeah, in a knife mouse brain. And what they eventually did is about, I don't know what that means for you guys. It's about over a maybe 10 billion photons per centimeter per second. That's kind of.
So you're telling me that that.
Oh, sorry. I think I'm wrong. So actually, it's about like a 1 billion photons. That's a lot.
So you're saying that constantly all our neurons are exposed to light?
I don't think.
Or are they shining?
First of all, they have the light influx at the surface of the skull, or maybe the brain itself, because they have to insert the probe. So basically, the skull part of the skull has to be removed. And then there is the difference between what happens in the surface and what happens in the POA. Let me calculate it by my mind. It's probably about 300 versus 10. So that is, 3,000 versus 10. So it's about like 300. So that is about 301, right? There's a little bit of light there. So
this is with very bright violet light,
should be very bright.
Are they using like sunlight?
They're using plasma light source, so it's very strong, probably like a laser thing.
Okay.
Yeah, They also have a microscopic adapter, so it's the light can be focused.
What about the fluorescent light bulbs that are in the animal facility? Would that get to the POA?
First of all, they have to get through the skin, right? So right now they actually, so in their sublunar figure, they remove the skin.
No, well, first of all, they have to get through the 3 cages that are stacked on top of them in the rack.
Well,
anyways, I think it's overall it's a pretty interesting paper, and they've used the right experiments and right techniques to address some of the basic questions. Any other comments? We're good?
Yeah.
All right. All right, I'll see everyone on Tuesday at 8 p.m. That's going on next week. Who's presenting next week? I guess not Alex.
whatever.
I'm amazed at how expressive your eyes can be.
It's exactly what my sister told me. It's like, how can your eyebrows move so much? Like,
right. Well, maybe
have
by
to.
Monday we'll decide who will present, depending on what we're seeing. Okay. All right. Thanks, everyone. Thanks for presenting.
Thanks.
And Amanda, we'll see you on Monday.
Yes, noon.
Okay. Yeah. All right.
Will you just send me like the address again? And then they said something about me having to go to the wild card office to get a special wild card with my fingerprint on it.
Oh,
Do I need that to get in, or is that just for like
I
the
have
animal
to do
stuff?
that too. If you want to come in on Monday, we can probably sign the paperwork or like take it together just so that we go in rather than, yeah.
Can we
do it
still
together.
get in?
Yes. So,
The
the
biometric
finger.
is just for the animal facility, right?
Okay, that's what I
Perfect.
wasn't sure.
Yes.
Okay.
So
Yeah.
if you can't get in, just Slack us and somebody will come out to get you.
Okay.
Probably me. But first of all, try. I think you can get at least into the building.
Alex, can you also
Yes.
Amanda card access and keys?
Yep.
I emailed Anthony and he said he'll look into it. So maybe there's another one coming his way. He'll do both at the same time.
Sure.
Thank you. I will see you on Monday.
All right.
Yeah.
Monday.
Actually, I have a question for everyone, for the new ones. Actually, did you all have to take the COVID training? Yeah. Amanda didn't.
Well, it's.
Does she? I think she would have to.
I think everybody had to do it. It's a training. It's like a video or something that you read and then you signed.
Oh, yes.
You
I did
acknowledge
that.
all of it.
Yeah.
There's no
Amanda,
test or anything.
Okay.
Yeah, it was
Everybody
just
did.
on Caesar. Like I couldn't sign up for classes till I did it.
Yeah.
Exactly.
Okay. All right. That's good.
Yeah.
I think on Lumen I can see that everybody did it.
And is everyone using the amazing symptom tracker app?
I keep forgetting.
Yeah.
The app is only available for Apple users for now. The phone app.
Online?
But everybody else, I think, are doing it online.
Okay. All right. It's onerous and it's hard to do that every day, but you know what will happen if one of us tests positive? They're going to say, why did you come in without having reported yourself? Oh. So this is mostly for the administration to cover their asses.
I'm free now.
But, they are not like making
try to do that and schedule it into your calendar so that you do it every day.
So we put it in the Lab COVID calendar.
Yeah. I already put it there.
So, yeah. And it's actually got really easy to do it. You just have to click 2 no's and then submit. So it got really better.
Yeah.
And
So you just have to do it for the days that you're planning to be on campus, right? Or every day?
yes,
Yeah.
just when you're coming.
Okay.
You don't have to do it on Sundays.
Okay.
All right. Take care, everyone.
Yep.
Yeah.
Bye.
Oh, hello. I just wanted to ask you like what your plan is for me for this quarter. Like I'm very flexible if you want me to just like slice things and stuff like that, or if you want me to like do a mini project with one of the grad students. Like I just didn't know what you had planned for me. I'm really okay with anything, but just having a better idea of like what I'll be doing.